First of all, thank you for inviting me to give this presentation on my research work. So I'm gonna talk about the on-chip gradient generation on cell cultures, and that is my PhD work, and the preconcentrating chemicals, which is my postdoc work, both relate to microfluidics. And so currently I'm living in Minnesota, USA. And so this is part one. I'm going to talk about my graduate work, which is most of the like 4 years of my graduate work in Texas Tech University. So in there, I focused on mainly on generating chemical gradient across a cell culture array of 256 cell cultures in a single chip. And then the part two, I'm gonna focus on my postdoc work, which is again using microfluidics, but different techniques, which is called isotachophoresis. So isotachophoresis I explain in detail when I'm talking about the research, but basically it's about preconcentration technique using different buffers. And so in my graduate studies, basically, since I focused on the gradient generation, bit of background about why the biomolecular gradients are important, because it's always important in the development, immune response, and wound healing, embryogenesis, and also cancer metastasis in most of the biological processes in our body. So specifically, I focused on like cancer cell therapeutics and finding drugs, what are the problems that face these days. And so to be more effective, to become an anti-cancer drug, it must penetrate through the tissues efficiently, as well as that it should target the population with the desired concentration to get a exert sufficient to exert a therapeutic effect. But the problem that they were mostly, even right now, they're facing is targeting unique molecule with desirable efficacy. That is a problem in drug discovery, because it's time consuming and labor intensive and expensive. So that's why the microfluidics comes to play an important role compared to the conventional method, because microfluidic gradient generators has specific unique features than the conventional methods towards the gradient generation on cells. And then because cell diameter usually range from 1 to 100 micrometers, and then also intercellular cell signals that secretes across gradient of length of 250 micrometer. But non-microfluidic platforms usually range from millimeter to centimeter scale, but the microfluidics always range from submicron to micrometer side channels. So that's why the microfluidics are more convenient and more has unique features compared to the conventional method, like precision, quantitation, and it has we can create spatio-temporal gradient control and than the traditional method. So there are several advantages using microfluidic gradient generators than using the conventional methods. What are the conventional methods that we know is that is the 96 well plates. So that gets like, if you want to test different concentration, you can only test like 96 concentration at a time. And also it's more of the fluid consumption, and you need a lot of yield and drugs to test on cells. So the merits of microfluidics is that cost of manufacture is really less compared to the conventional methods. As I said, it's reduced the reagent consumption because we need very small amount, like microliters level, and faster in analysis because we can even test several concentrations, or several tests can be done within 1 hour or less than 1 hour of time. And it's increased the separation efficiency, and also because of the compatible material that used in most of the microfluidic devices that we developed and designed, it's well suited for cell analysis. And also it's increased the portability because it's pretty small devices and you can go and test on site, and it's point of care diagnosis. It can be used for point of care diagnosis, and ease of use and disposal is another merit of microfluidics. So this is the design of the gradient generator or the culture array chip that I designed. In this chip design, you can see that it's a Y shape, so you can flow one fluid from one side and the other fluid from the other side. So there is two layers, which is called bottom layer and the top layer, and there's a main fluidic channel, and then the bottom layer is the vacuum channels. So we use the vacuum actuation to load the fluids into side chambers, and the main fluidic channel is the top layer, so that's called the fluidic layer. And this is the zoom in device, like part of the device, the beginning of the first part of the device. So you can see it's a V shape. Then you can mix the solution, and because of the diffusion, it can create the concentration differences. I'll explain that later on. And for this device, we use the PDMS, which is the polydimethylsiloxane. And there are several advantages of PDMS, using PDMS, because it's optically transparent and its biocompatibility and also permeability helps to load, keep the cells survive because it's gas permeable, so the oxygen will be supplied through the device, and it's incompatible with many organic solvents, and also it's elastomeric, so it allows, once they actuate, it has this reversible deformation, can come to the same design that we had before. And also the reactivity, it's really unreactive towards most of the region, it's very unreactive. And then surface can be etched, so we can modify the surface as we want, and also can be modified to be hydrophilic because it's mostly hydrophobic because it's an organic molecule. So for the developed design of the chip, we use the silicon wafers as the mold. So then once we have the device ready, I did the simulation and experimental validation. Using the simulation, I did the collaboration with the mechanical engineering department and mathematics department in Texas Tech. So they used the COMSOL multi-physics for that. So this is my second publication. So initially we thought of having like this side chambers is 150 millimeter long, but then it takes some time to get the diffusion into those side chambers. So to minimize more and get more faster analysis, what I did was like decrease the length between the V shape and the starting point of the side chambers, and also decrease the side chamber length. So that kind of increased the efficiency of this gradient generator by decreasing the time that it takes to stabilize the gradient as a function of distance. So this is some illustrator diagram that shows that how that one inlet having one solution, the other inlet having a different solution. So when it's flow, the laminar flow, and then it's kind of diffuse into each other and create different concentration as a function of distance. So the if I talk about the fabrication, basically I use the soft lithography technique. So that's what my group use most of the device fabrication. So in the device fabrication, you use the negative photoresist, the SU8, and then you do the spin coating, and you do some soft baking, and then expose the mask. So mask has the design that we want. So you use that, we design using AutoCAD, and then get the mask from some suppliers. So then we do the UV exposure. So it's kind of etching your SU8 photoresist, and then do the post baking to make it hard. And then you do the developing to get rid of the excess SU8, and then dry and cyanize, and you develop the pattern. So once we have the desired pattern, then using the PDMS, we do the fabrication of the chip design. And then, as I mentioned, in my chip design, there's top layer and bottom layer. So top layer is the fluidic layer, and the bottom layer is the vacuum layer. So for the top layer, we use 1 to 5 curing agent. So curing agent is to use for the PDMS to make it hard, because PDMS is like a slimy material, and then to make it hard, we use the curing agent. And then for the bottom layer, we use the spin coating, but the ratio between curing agent to PDMS is 1 to 25, because we need very thin layer for the bottom layer, since we use that for the vacuum actuation. Then once we spin coat, and then we bake at 70 degrees, because we need that thin layer. And then for 30 minutes, after 30 minutes, we paste the top layer, which is the fluidic layer, on the bottom layer. So that should be done really carefully, because you cannot, you have to be really careful with the pasting on the right position. That's the most critical part in this device fabrication, but once we get used to it, it's easiest task. So then we bake that for 120 degrees for 3 hours, and then finally we add the tubing as we want, and then get the final design, and then we do the plasma sealing to the glass slide using oxygen plasma. So that's the device, but there's no zoom in device, but this is the chip design. So it's pretty portable and pretty small in size. So, and then how the device works is that, since mostly I use that for the cell-based studies, and then side channels would be filled with the cell media, and or then you can flow the dye or drug from one inlet or buffer or cell culture media based on what you do for the experiment. And then for the demonstration, I use the fluorescence dye how to show like how the chip works. So then fluorescence dye can be flow from one inlet, and buffer, if I use the cells, we flow the cell media or buffer. And then when you have fluorescence dye come from this inlet and the buffer or cell media coming from the other inlet, then it's diffused into each other. And then I refer these channels as upper side channels and lower side channel for the lower one throughout my presentation. So when diffusing to each other, initially you have high concentration of the fluorescence dye in this side for upper side channel, and as a function of distance, that concentration is decreased. That's what shows from these color differences here. And then we can detect those using fluorescence microscope, and the fluorescence microscope, like you see here, so we can set up the flow from different injectors and then let it go for some time, and then get the stable gradient. So if we use the chip for the cell-based study, and then this is the design. So you have vacuum channel, And if we apply the vacuum, so this video shows once you apply the vacuum to the vacuum channel, then these cells can be loaded into one side. So if you apply the vacuum to the upper side channel, and then the cells will be loaded into the upper side chambers, and then we can load the cells into the lower side chamber by actuating the vacuum here. So it's then cells can be stay there for a long time, and I will show you the cell viability. We did a long-term cell viability study. So once the cells loaded, then we can use any drug or any chemical that we want to test it on cells. So yeah, that's how the cells loaded, and then even for the fluid loading, we do the same thing. We use the vacuum actuation. So for the validation of first we need to validate the data, because before we use into the cells analysis, we need to make sure that there's no shear stress on cells. And because when you have high shear stress on cells, if there's any fluid flow into the cell chambers, then cell can get a more stress on cells and they can die. So in order to do that too, and also to validate other data, we use the simplified Navier-Stokes equation for creeping flow, and then fluid flow in pressure. I'm not going to talk about a lot about the mathematic part here, because I more focus on chemistry, and this is done by the collaboration. So fluid flow in pressure driven microfluidic, that's governed by Stokes equation. And then, so there's a pressure gradient, and then the dynamic viscosity counted and the velocity. So, but the diffusion of dye distribution is mostly based on convection diffusion equation. So these are the differential equation we use to do the validation using COMSOL multiphysics. And in the main channel, there's the convection part as well as the diffusion part. But when it's come to the side channels, only count the diffusion part, because it's being shown from the simulation and the validation from the COMSOL. So we found out that mass transport into and out of the culture channels occurs mainly via diffusion with a small convection component. That's why we don't have convection component for the side channel here. We only count the diffusion to the side channel. So to demonstrate how the chip works, I use the fluorescence dye as I mentioned before. So, and then flow the buffer from other side, and then once we have two flows going into the channel, you can see that in the beginning of these are the intensity values. Beginning of the channel, upper side channel has higher intensity value, and it's keep decreasing towards the end, which means that concentration of the dye is decreasing. And then opposite happens for the lower side channel. Beginning you have lower value, and then as a function of distance, it's giving higher intensity value. So that shows us that initially you have higher concentration, and it's keep decreasing for upper side channel, and then keep increasing for lower side channel. So we have different 256 different concentrations created at the same time in a single experiment. So to validate results on and do some experimental application on cells, first I use the ethanol gradient on cells, because that's the easiest one to make sure that how the cells respond. And then using the ethanol gradient, cells were stained with propylium iodide, and then did the viability study. So you can see that the lower side channel, the viability is higher in the beginning, but at the end it's decreasing. But upper side channel, the viability is pretty low, it's 0, because you have high concentration of ethanol at the beginning, and then it's keep increasing the viability as the concentration of the ethanol decreasing. So this kind of indicate that this chip can be used for cell analysis too. And then we use the mathematical model and simulation results to validate the fluorescence dye data experimentally found, and then also using the simulation. So we input all the parameters that we use in experimental to console multi-physics, and then console created the simulation, and you can see it has the same trends when you have concentration calculated for dye and then concentration calculated using simulation. It has the same trends. So there are some fluctuations here like this. That's because of the photo bleaching, because we didn't have automated microscope that time, so I had to take like 1 or like 6 or 5 chambers at a time. So, but nowadays we have like automated microscopes that we can minimize the photo bleaching of the dye. So this is again mathematical the validation. It's like 2D distribution of the concentration inside domain of the flow. So you can see that there is no the convection into the side channel, and this experimental and simulation results show the same. And then we use the concentration values of the dye vertically cross section inside different channels. So you can see from the 5th channel to 120th channel, and in the 5th channel for both sides, the concentration difference is higher, but when it go down the chip, the concentration difference getting lower. So that's again confirm the mixing and the diffusion and creating the different concentrations. So to make sure that there's no velocity profile or no fluid go into the side channel by convection, then we use the console and calculated the velocity profile of the domain of the flow, but we can see that there is no fluid moving to the side channel. It's only via diffusion. So that also shown in the experimental image. So while flowing the cells, I took this image. So it's only have high velocity in the main fluidic channel, not into the side chambers. So that's the positive part of this one, because there's no shear stress develop on cells evenly flow at high flow rate. The medium is flow at high flow rate in this channel, so we can keep the cells viable for a long time using this chip design. And then because it's validated through the mathematical simulation, I wanted to do the experimental validation and then calculate the shear stress actual values. So then this is how the fluorescence dye diffuse into side chambers. And then once we calculate, then usually the cells can only survive under shear stress between 2 dynes to 100 dynes square centimeter per square centimeter, but we calculate the shear stress inside the side chambers at 0.04, which is pretty low compared to the cell survival range, and then in the main channel is 1.3. So that is the advantage of this device, that the shear stress is minimized on cells. So then I did the long-term culturing and showed that even adherent cells and also suspended cells can be viable for 96 hours with a 90% viability. So we can grow cells inside the chambers, even adherent or suspended, with a 90% viability for 4 days. So that is really big advantage, because you can grow cells for a long time and then test different concentrations on the cells and see what is the lethal concentration of the drug, and we can target more with a efficient therapeutic effect. So, but we have to make sure that the gradient is stable for longer time too. So then I did experiment with the longer chambers. It's take 45 minutes to stabilize the gradient. And then I again tested after 24 hours. So you can see it's giving a stable gradient even after 24 hours. So once the gradient is established, the consistency does not change. So it's very consistent results even after 24 hours. So that is another important factor. And also flow rate. So when you change the flow rate, you expect that the gradient profile will be changed. So I increased the flow rate from 0.06 to 0.08. And then you can see that the gradient, we can create different concentrations. So within the same single chip, and also it's validating using simulation gradient profiles. So this is really a big advantage, because once we can create 256 at one experiment, and then again, using different flow rates, we can create more of different concentrations, which is really important. And these gaps between these lines, that's also again due to the photo bleaching effect. So I kind of avoid those values to get a more consistent results. And then with the shorter side chamber that I said, like we use the shorter side chambers for the further analysis with 0.75 millimeter. It only takes like 30 minutes, so it's make it faster analysis. So even after 1 hour, you have the same gradient. So for the testing the application part of this chip design gradient generator, I used the Ramos cells stained with calcium AM and used the H2O2 gradient. And then you can see it has some errors because these are cells, and cells like behave really different from each other. So there's kind of having some errors applied. But the gradient works well because with the H2O2 gradient, there's a concentration decreasing and the viability. The medium side is the lower side, and then the concentration increasing for medium side, and then viability decreasing. And for the H2O2 side, the concentration is decreasing and viability is increasing. So this is shown in the picture here. So when your cells are alive, the calcium AM is a fluorescence dye, shows as green under fluorescence microscope. So it's in channel 9 and 10 at 0 hour, but after 0.75 hour, the viability is decreased. So that is in the H2O2 side. And then for the channel 81 and 82, H2O2 side 0 hours, the viability is higher, and then it's increased to 82%. Not increased, but it's not decreasing as the previous or the channels that found in the beginning of the H2O2 side. So this is also showing that we can use this chip for analyze different concentrations on cells, and to make sure that how the conventional gradient and microfluidic gradient works together, we did the conventional analysis. That means we use some Petri dishes, 6 Petri dishes, and use the same concentration calculated from the chip, the gradient generator, and then apply those concentrations. And you can see the cell viability in Petri dishes and cell viability in culture chambers, giving a linear graph, with the R2 is 0.98. So that again shows that the calculated concentration using the mathematical models are accurate, and then we can use that for further analysis. So what we achieved in this 4 year of studies is that 256 different concentration can be generated within 20 minutes in a single chip with a simple operation, and also can be used for long-term culture under low shear stress environment. And long-term gradient can be created with minimal gradient consumption of 1 milliliter. We only take 1 milliliter of the sample to test it on. And the concentration values in each side chamber can be calculated using mathematical model. So that's what I achieved from my PhD work. And then I started my postdoctoral work at Department of Mechanical Engineering, Texas Tech in 2016. And there mostly did a collaboration work with the documentaries in UC Berkeley, and they were using this universal chemical analyzer to study chemical or the fluorescence dye called Pacific Blue Succinimidyl Ester, which can be attached to amino acids. So then they were using this universal chemical analyzer in the mass organic analyzer. So they have this project going on with NASA, and then so if there's amino acid that can be detected in mass, so that they kind of want to know whether there's any lives in mass. So that's the main project. But when you do such experiment, that we can collect only very small amount or trace amounts of molecule. But when you have trace amounts of molecules or the chemicals, then detecting those under microscope or any other analytical technique is more difficult, because we need at least sufficient amount of the concentration. So that's why we use this isotachophoresis, that even when you have trace amounts, this is a non-linear electrophoresis. And in isotachophoresis, what it does is pre-concentrate. So it can be pre-concentrate like trace amount, like millions times. So instead of single electrolyte that we use in electrophoresis, isotachophoresis, or I can say ITP, use discontinuous electrolyte. So there's two coions, has different mobilities. One is have a higher mobility and the other one has a lower mobility. So we call leading electrolyte for the high mobility one, and the low is mobility one as trailing electrolyte. So LE and TE. So when you have a sample in between that, the mobility of the LE and TE, it can sandwich between those two electrolytes and then pre-concentrate. So what happens is that your trailing electrolyte goes later in the channel and leading electrolyte will move forward. And if your sample is in between those two mobilities as electrophoretic mobility, it can pre-concentrate in between those two electrolytes and get purified samples on. So what happens here? Pre-concentration of trace amounts. So in here, that ions A, B, and C, it has different mobilities. So A has lower mobility and B has in between A and C. So this method has a self-correcting mechanism. So it goes in the same velocity, also the same velocity, but if their electrophoretic mobility is different. And when you apply electric field, when you disturb isotachophoresis conditions, then it can still self-correct the mechanism. That's the main advantage of this method. So what I did in my postdoc study is the characterization and optimization of ITP for parameters for PD. We call Pacific Blue Succinimidyl Ester dye, because it hasn't been using ITP. So we have like single channel chip device, and then when you apply the electric field with the different buffers, so you have sample inlet with the TE buffer, and you have LE valve here. When you apply the electric field, then your LE will lead the solution, and then TE go after the sample, and then you get a PO sample zone. So in this case, I found out that the best two buffers are the 203 HCl, because HCl has chloride ions, which is pretty ionized, and cation, and then at pH 8. So the pH is important too. So I did many studies with a different pH and found out the pH 8 is the best option. And 40 millimeter sodium tetraborate at pH 9, that is used as the LE and TE. So you can see here that at T0 seconds, there's no dye detected because it's not able to concentrate. But with the time goes by with the function of distance, then you can see that the at T equals after 60 seconds, after 1 minute, it's pre-concentrate. But after that point, it's kind of getting diluted. But when you collect this one at some point, then we can detect that sample even using mass spec or HPLC because it's pretty concentrated. And here the video that how this band is flowing. So it's flow that in the same velocity, and then you can detect under the microscope. So the pre-concentration ratios were calculated, and you can see then the initial concentration is 0.0001. The pre-concentration ratio is increased to 20,000. So, and 1 point it's because it's high concentration of nanomolar, but we can detect even pretty small nanomolar concentrations using this technique. That's the beauty of this technique. And then with another grad student, I used the fully integrated isotachophoresis to using programmable microfluidic platform. So it's pretty automated. So you have the ITP channel attached, and then we use the vacuum actuation and fluidic layer and the pneumatic layer. So there's 3 layers: pneumatic layer, fluidic layer, and glass wafer. So it's a programmable microfluidic platform. So you have this DAQ switching board and electric feed connected to the ITP channel, and then you have power supply. So then using this automated system, they can actuate the valves in this system, and then you can even use that for concentration studies. So in device operation, what we do is first we do the priming using the LE buffer, and then we load the sample and buffers, and then run the isotachophoresis and wash the channel, and again we load the sample and run. So we can keep doing that because it's automated system. So priming is you need to have filled all these valves and all these the device with the LE buffer, and then you apply the loading the TE and the sample into the ITP channel. Then when you apply the electric field, it count the TE buffer coming from this side, and then the LE coming from this side, and then it form the pre-concentrated band. So this is the picture, we use just the dye to show how this works, and then this is how the process of PMP, and then we use three different analytes or 3 different dyes at the same time using 3 different buffers. So borate and HEFEs and phosphate, and the borate is the one gives the best data compared to the other two buffers. And then this is PB, this is the Pacific Blue Succinimidyl ester, and Analyser and Analyser 488 has different fluorescence wavelengths. So we use this one device, and we had the samples in the same sample, and then when we apply the electric field, you can see how it's pre-concentrated, and it could be separated. We can separate because the electrophoretic mobilities are different. So the future work, this gradient generator can be used for the one that I introduced for drug discovery, for cancer, and also this automated microfluidic platform can be used to provide pre-concentrated samples for capillary electrophoresis, mass spectrometry, and many more analytical instrumentation. And then all these microfluidic devices that I introduced are very useful in vivo studies. And I would like to thank first for inviting me for the METMA, for inviting me for this during the seminar. And also this is my PhD advisor, Dr. Dimitri Pappas, and Dr. Akif is the collaboration mathematical simulation, and Dr. Shehan Habitanti, my colleague, he's the one who did the simulation part, and the Pappas group at that time, and Dr. J. Kim as the postdoc advisor, the BMND group, the mechanical engineering, and my family and friends who all supported. And thank you all for being here to listen to my talk too. And yeah, this is one thing, some photos from Sri Lanka, and because I graduated from Sri Lanka and then came to Cal State and Texas Tech, Augsburg, and now working as in Gustavus. And thank you very much.